So hello everyone and welcome to the 21st I3 session on PGTA in the lab. I3 is the International IVF Initiative, and it's our honor to provide you during these unprecedented times with a platform for continued education and collaboration, help you stay busy, productive, in touch with the IVF community and lab scientists worldwide, through webinars, online materials, in an array of relevant topics. So we're happy to have you all join us, and happy to have everyone tell if this is real. So if you've been following me on social media, I got bored and let my hair go, and then I got really bored this weekend and we made a mohawk. So that's real. I do have hair, even though I've been shaving it for 20 years. So keep the comments about the hair coming. It's not colored though, but you know, maybe later.
Platinum's in style now, don't worry. Okay, hello everybody. My name is Shaista Sadruddin, and it has been such an honor and privilege to be a part of this fantastic organization. We would like to encourage all professional organizations, if you haven't already done so, to join this initiative and partner with us, like several local, national, and international groups have done. As a reminder, we are a neutral organization with the prime mission to serve you, the reproductive lab community. So please let us know which topics you are interested in and how we can be of help. All of our webinars are recorded and posted on our website should you want to revisit them or if you have missed any previous sessions. We have already presented a couple poll questions for you to gauge how people are currently using PGTA and biopsy techniques in their lab. Again, there are 2 questions, so please be sure to scroll through and read all the questions and answers before submitting. I would now like to introduce my co-host, Dr. Gary Harton. Gary got his PhD from the University of Kent, where he studied facilitating the widespread use of pre-implantation genetic diagnosis and screening through best practice and novel technology development. After over 20 years serving in various roles, including both clinical laboratory and management positions, Gary began his commercial career performing business development and market development roles with 2 prominent genetic companies, a fertility startup and a large reference genetic laboratory. Gary currently resides in Virginia, right outside DC, with his wife, his 5 kids, and 2 very large and lovable dogs, and potentially a 3rd one on the way. Is that right, Gary?
Yeah, something like that. One of them's in heat, so we're not really happy with the dogs right now. Thanks, Shaista. I'm really happy to be one of the hosts here and moderators for today's session on PGTA and a speaker. So I'm pulling a few duties today. So that's really exciting. So I'm virtually sitting next to Shaista Sadruddin. I wish someday we'll hopefully meet in person, but we're spending a lot of time on Teams together. So the 2 of us are going to work hand in hand over the next hour and a half or so to host this presentation, ask questions to our guests, maybe answer a few questions ourselves, hopefully. Before we get started, I want to tell you a little bit about my co-host Shaista. She graduated from the University of Georgia, which she had to explain to me when she said she went to UGA, that what University of Georgia, that UGA was, with a BS in biology, and recently graduated from Eastern Virginia Medical School in May of this year with her master's degree in reproductive clinical science. Her thesis was titled Analysis of the Validity and Consistency of NGS Testing on Mosaic and Aneuploid Embryos. Shaista works at RBA in Atlanta, Georgia as an embryologist with Peter Nagy and his team. So please note that all questions to the speakers must be asked through the Q&A feature, so they can be sent to Shaista and me to be asked ahead of the speakers. Please do not use the chat function. We will not be checking there for questions. Submitted questions will be answered either verbally by the speakers or in writing by the panelists after we close the session.
At the moment, we have over 1,400 people registered for today's session on PGTA, which is not surprising since this continues to be a very hot topic in the IVF art field. We will be spending the next 70 to 80 minutes discussing how we can improve PGTA outcomes and techniques from a laboratory perspective. As Gary said, we encourage all of you to use the Q&A function to ask questions. We will try to address as many questions as possible during today's session. The remaining questions will be posted on our website after the fact. At this time, we'd like to display the results from the poll that we had asked you guys about earlier. There we go. So according to this poll, what biopsy method does your lab primarily use? 74% of us use laser technique, which is like I believe what I use in my lab too. 13% use the flick technique, 5% said other, and 9% said non-applicable. The second question is, when performing a trophectoderm biopsy, when do you breach the zona pellucida? So we have 4% that have said either day 1 or day 2, which is prior to biopsy. 37% do it on day 3, 13% on day 4, 11% the morning before biopsy, and 25% the moment of biopsy. And we have 8% saying other. Those are some very interesting facts. Today we are joined by some truly distinguished guests who will each be giving a lecture followed by a few minutes for Q&A. On our panel today, we have Dr. Gary Harton himself, Dr. Peter Nagy, Dr. Dawn Kelk, and Dr. David McCulloh. We are thrilled to have them on and to contribute their expertise on the subject of PGTA. Their disclosures of affiliation can be found on our website in their biographies. We will begin with my very own co-host, Gary Harton.
Thank you very much, Shaista. I was really happy when Peter and Jacques asked me to start out the webinar today with a brief discussion on the nuts and bolts of next generation sequencing and how we use NGS in the lab to perform PGTA. In my script that I'm dutifully following, I made sure to highlight and bold the word brief because Dr. Cohen has told me on a number of occasions how I tend to go on and on when I start talking about genetics. So I'm going to try and do my best to be brief. So if you will bear with me for just a moment, I'm going to share my screen and then we can get started. Okay, Shaista, can you confirm that the slides look right?
Yes, they look perfect.
Okay, great. All right, so again, the title today is Next Generation PGTA with NGS or Next Generation Sequencing. Here are my disclosures. They're also on the website, so I'm not going to belabor those at the moment. I think it's important to start this, anytime we talk about PGTA, with this sort of brief foray into the past. There's a lot of names that this technology has been called. Pre-implantation genetic screening, or PGS, is the one that you're probably most familiar with. There was the use of comprehensive chromosome screening, which sort of took hold a few years ago when some people wanted to sort of separate themselves from doing PGS, perhaps. As a matter of fact, I was just reviewing abstracts for ASRM today, and someone's still using CCS. There's other names. I have a few here: aneuploidy screening, whole chromosome screening, etc. In 2016, the World Health Organization harmonized those terms to the following. And we need to stick with this because the more we use different terminology, the harder it is for everyone to keep up with what's going on. So we're talking about pre-implantation genetic testing for aneuploidy or PGTA, right? That's what it should be. So it should be PGTA always. And I also like to just sort of review a little bit about the PGTA, the evolution of PGTA, because I think it's important to understand where we came from and where we are. Certainly in the early 1990s into the mid 1990s, we were using multicolor fish to look at a few chromosomes. If this has been termed sort of first generation PGTA, I think Santi Monet is the one that sort of started calling newer methods PGTA 2.0. Jacques Cohen did point out to me that there's sort of a hybrid in between here. There were a couple of groups that were using fish but doing all 24 chromosomes. But for the sake of simplicity, I like to think of it as multicolor fish. Even for 24 chromosomes, it was really difficult. That's first generation PGTA. And then everything since then, which includes metaphase CGH, array CGH, qPCR, SNP arrays on here, and anything to do with next generation sequencing NGS, I think is sort of second generation PGTA, because we can very easily and concisely look at all 24 chromosomes at one time. And that's the hallmark for me of PGTA 2.0. So to go from there, let's talk a little bit about next generation sequencing. And so this is Jim Watson, he of Watson and Crick, holding a hard drive in May of 2007. That hard drive has Dr. Watson's entire his entire genome is embedded on that hard drive. So that's the basis from the top of chromosome 1 all the way through the bottom of chromosome Y and every base in between. It's about 3 billion bases. Jim Watson was one of the first 2 people in the world that had their entire genome sequenced. J. Craig Ventnor was the other one. He was the guy that owned the company that did most of that sequencing. The reason I'm telling you this is that back then, in 2007, that project took 15 years to complete and cost about $3 billion to get 2 genomes, 15 years, $3 billion. The reason we're talking about next-gen sequencing today in IVF and in PGT is this exact reason. Sequencing companies today can do that same exact trick, sequence your entire genome for about $1,000, and it takes less than a week to get done. So like most things with any technology, we've gone from 15 years and $3 billion to 7 or 8 days and $1,000. And it's just going to keep getting cheaper. And so I think it's not only going to be PGT that you see sequencing in, certainly if you look at the COVID things that are going on, it started with PCR tests, then serology tests. Now we've moved on into next generation sequencing tests to look at where where the virus came from, et cetera. So next gen sequencing is creeping into every bit of medicine as we see it. And so I think to talk about next gen sequencing, we really need to start at the most difficult part of all this, which is the biopsy, right? I can't underplay how important it is to do good biopsy and to handle the sample really, really carefully and well until it gets to the PGTA laboratory, and that's going to create the best results. And that's what my co-speakers are going to talk about today, is things that go on in the lab that might affect the outcomes. But before we do that, we're going to talk about how we actually get a result. So you do a biopsy, we get a few cells. In those few cells, there's a little bit of DNA. There's not enough DNA in there to do anything really fantastic with. There's certainly not enough DNA there to do next generation sequencing and get a real result. So we take the embryonic DNA and we do whole genome amplification, or WGA. And there's a million ways to do it. I'm not gonna talk about that. But let's just presume you do some sort of amplification, WGA or a targeted PCR. And so what happens then is you end up with then lots of DNA. And now you want to get that DNA ready to go on the sequencer, right? So you take your unknown DNA, you fragment it down to a certain size, and then you need to put what's called adapters on there. And those adapters are nothing more fantastic than little pieces of DNA that each sequencing system uses to move the DNA in and around the sequencer, because DNA doesn't just go into a tube and get sequenced, it goes onto a hard platform. So adapters are simply pieces of DNA that are used to move DNA around. So the amplified embryonic DNA is fragmented into smaller pieces, and then adapters are put on, and you have then what's called sequencing ready library. So you then load all that DNA onto the sequencer, and the sequencer does what? It just sequences. It sequences every region of the genome multiple times, over and over and over again, right? So the typical sequencer that you see in a PGT-A lab globally today, you average about 25 million reads, meaning that there's 25 million fragments of DNA that are sequenced, right? And so we have to do then something with these literally millions and millions and millions of short sequences. So the first thing we have to do is align them. We have to figure out, where does it belong? So in the end, there is a human genome. It's called the HG19 genome. It is the one genome for all humans, even though we don't all match up to it exactly. But there is one reference that we all agree to as the reference. And so you take each piece of DNA, you notice the orange and blue adapters on the ends. The sequencer forgets those and just reads the gray bar down the middle, which is the unknown sequence, and it aligns, right? It says, where does this piece of DNA belong in the genome? Does it belong on chromosome 1 or on chromosome 6? And it assigns it then to which chromosome it belongs on, but not only just which chromosome, but where on the chromosome it belongs, which means it might be the first piece might be on the P arm, the very top arm of chromosome 1. The next piece might be right near the centromere on chromosome 8 or wherever else. And so that's what's called alignment. Now, if we did what I just described, for each embryo, it would, number one, take forever, and sequencing will be quite expensive, because obviously there's a raw cost to do sequencing. So some really clever people came up with this other idea called barcodes, right? It's not unlike the barcode that you use when you get on an airplane that says this is, this is Gary Harton's seat. He's sitting here and he's meant to be on this airplane going to wherever. Or if you go to the grocery store and you buy a loaf of bread, the scanner knows exactly how much that loaf of bread costs. So we use DNA barcodes, which allows us then to combine multiple embryos into one sequencing run and multiple embryos from multiple patients into one sequencing run. There, then, you're taking advantage of this 25 million reads. You can spread those across hundreds of samples, right? Depending on how big your sequencer is and what your throughput needs are. There's 8 sequences all the way up to hundreds of samples, maybe even 1,000 samples on a much larger sequencer. And so then you go adapters on the outside, because we still need to move the DNA around, barcodes next, then the unknown piece of DNA in the middle. So then we do the same thing. We make libraries, put the barcodes on, sequence. And then when we do the alignment, not only are we figuring out where in the genome does this piece of DNA belong, is it chromosome 1, chromosome 2, chromosome 3, but also where did this DNA come from? Is this Mrs. Jones's embryo 1 or Mrs. Jones's embryo 2? Or maybe it's Mrs. Smith's embryo 4, right? Because we can combine multiple embryos from multiple patients into one sequencing run, and therefore really harness the power of next generation sequencing. So once you've aligned all of these pieces of DNA, you're going to align them and you're going to place them into a bin. And a bin is simply where we take the genome and break it down into usually approximately 1 million base pieces of DNA, and then you simply count across those bins. So you see here on the very top, those are the bins, and you can see there are various counts from different places in the genome, all along the chromosome, and then each of these bins gets consolidated down into a dot that you're typically used to seeing when you look at one of the profiles that a lab might show you, or that I might show you when I'm doing a talk. So you can imagine there's a lot of bioinformatics that goes into whole genome amplified DNA, sequenced, aligned, cut down into bins. Those bins are counted, and that count is represented by a dot somewhere along the chromosome. And that's essentially how next generation sequencing works. So once you've got all those dots, then the system compares those dots to something, right? So this is the way Illumina does it. Thermo Fisher does it very, very similarly. What they've done is they've taken some number of normal people, they've taken some single cells from those normal people, sent them through the whole process, whole genome amplification, library prep, sequencing, alignment, counting, binning, all that, and come up with a representative number of how many counts along each chromosome one should get for any one chromosome. And you can see here, chromosome 1, chromosome 2, chromosome 3, they all have different counts based on how many genes they have, how gene rich they are. And then the unknown is compared to the known, blue to orange here. And you can clearly see down here on chromosome 22 that there's way too many counts on chromosome 22. And so the system can then call that a trisomy, right? But again, remember that in sequencing, we're not only counting how many are there in each chromosome, but we're also counting where they are in the chromosome. So next-gen sequencing allows you really to get, pretty good resolution. Some platforms can get down into the 2 megabase, 3 megabase resolution routinely, whereas with array CGH, we're more likely at around 10 megabase resolution. So this makes chromosome rearrangements a little more interesting. You can see more segmental chromosomes. And because of the power of the fact that you're actually counting chromosomes, this has led to the ability to also see mosaicism, which we're not really going to talk about a ton today, but we're all aware of mosaicism and that it's a particular thing that NGS allows you to see. So you take those chromosome counts. In most softwares, you can drill down to the chromosome level. So here you can really see where each of those pieces of DNA, where those bins are assigned. You can see that they don't actually come out in these perfect nice lines that we like to show you. The real scientists in the PGT lab, this is where they go when they want to really look at a chromosome and see how it's impacted. But you can see, because of the resolution, the system then allows you to look at, for this is a translocation, a gain, or a loss on the arm of the chromosome, and you can see exactly where that gain or loss are delineated, either for a de novo segmental or in a translocation case. And then again, this is what that looks like when we show it to you. Of course, this is a good looking one, but if you look really carefully, you can see there are hundreds of dots along every chromosome. And every one of those dots is a representation of the counts from the bins from all that sequencing that we did. And then you can simply look across and see here very clearly, there's a loss, a segmental loss on chromosome 1 and a segmental gain on chromosome 11, simply because this is from a translocation case. And so hopefully in about 10 to 11 minutes or less, I didn't time myself, that's how next generation sequencing works. And I thank you very much for the time. And I thank you very much, Peter and Jacques and the rest of the i3 team for inviting me along today.
Thank you, Gary. That was a fantastic presentation. I'm going to ask you one question before we move on to the next speaker. It seems that some software are more prone to make mosaic calls. Do you think this increasing specificity in detecting mosaicism has a benefit? Can you describe the differences in technology that cause these variabilities in mosaicism detection?
Yeah, I think, Shaista, that's a really good question. And I wish I had another hour because, that's a whole lecture in itself. I don't know that it's the systems that are producing this. I think it's more to do with cutoff values, right? So a laboratory that might look at mosaicism and say, I think the lowest level should be 30%. Another lab might look at that and say, I think it should be 20%, right? It doesn't make one right or one wrong. It just simply means they're different. But obviously, if the low cutoff is 20% for one lab and 50% for another lab, that really means that a lot of people, there's more embryos from one group or another are going to be called mosaic. And I think for me, as a lab scientist, it's really easy for me to understand that and to really say to my customers or whatever, this is what our cutoffs are. I think the more difficult part comes when it comes to embryologists and nurses and doctors that have to explain this to patients and have to decide what to do with these things. I think ultimately what we have to do is all work together to, maybe we're never ever going to harmonize on what the cutoff should be, but we need to harmonize on how we see mosaic embryos, how we talk about mosaic embryos, how we talk to patients about mosaic embryos. I think really for me, that's where this question comes. It's not my lab versus their lab or my system versus their system. That's not what this comes down to. For me, it comes down to individual labs, how they use the system that they use and how they communicate that to their customers. I think that's the biggest thing for me. That's the big take home for me here. And again, I'm always happy to come talk about mosaicism because I think it's a big problem in the field for sure.
Absolutely, yeah, I agree with you. I think there's a lot to delve into there. Before I move on to the next speaker, I would like to pose one additional poll for you guys to answer. There are 3 questions on this poll, so please be sure to scroll all the way through, read all the questions and answers before hitting submit. And as you guys do that, I would now like to introduce our next speaker, who I have known for as long as I have been an embryologist. Dr. Peter Nagy is known by many in our industry for his numerous publications and talks on embryo science. He has also helped found North America's first vitrification-based egg banking program, and he was a pioneer in developing the ICSI technique that we now all know and use today. Dr. Nagy is currently the Scientific and Laboratory Director at Reproductive Biology Associates, RBA, here in Atlanta, Georgia, where he has the great pleasure of being my boss and mentor. So with that, I'll hand it over to you, Peter.
Well, thank you very much, Shaista. I really appreciate this introduction, and if I may just say that it has been a huge pleasure for me, to be part of our team here at RBA, and actually, I would just really say that regarding, for instance, vitrification, that has been really a team effort, and it was my colleague, Dr. Jeremy Chang, who has been leading, all the development on vitrification. So it is just a wonderful team, and you are part of that great team. So thank you so much. I also appreciate for i3 giving me the opportunity to talk about PGTA and also what is the effect, as well as the concordance after re-biopsy. So we all know that the goal of IVF is to provide infertile patients with a healthy baby. And obviously there are other points to consider, like how much time does it take, to get the baby, how much does it cost, what is the convenience, whether, the patient has to try one time the IVF or 2, 3, 4 times to get through an IVF stimulation cycle. It's also important, if the couple is desiring to have just one child, or if they would like to have 2, 3, or more children. So depending on all this, what patient wants, approaches may differ. And, we all know that embryo assessment, what we practice in the IVF since the beginning of IVF, is helping to improve efficiency of the IVF process. Now, the way that we do the embryo assessing is to try to identify those that are viable and contribute to a healthy singleton pregnancy after a fresh or frozen embryo transfer. We do that, traditional method with the microscopy examination of the embryos. However, this approach, as we all know, it's not very efficient, not very reliable. And that is the reason that, our community is looking for additional tools for quite some time. And, one of the best known and applied tool already for decades also, what we call the PGTA, or how is it called earlier, PGS. This tool is looking at the chromosome composition of a biopsy sample and able to identify viable or non-viable embryos based on that test. So let's just give an example here. A patient who has 4 high quality embryos, morphologically they all look very similar. And let's assume that if this patient has a 50% implantation rate with these 4 embryos, this means that if you transfer all 4 embryos, then at the end, the patient will have 2 babies out of these 4 embryos. Let's also assume that the aneuploidy rate for this specific patient is 50%, which is quite usual to see a 50% average aneuploidy rate. If that is the situation, and you perform an embryo biopsy, and you deselect those embryos that were aneuploid, so you have 2 euploid and 2 aneuploid embryos, you remove the aneuploid embryos, and now you are using only the euploid embryos, and you transfer only those 2, then you should also have the same 2 babies out of it. And that means also that your implantation rate now should be increased from 50% to 100%. And that's where the efficiency of PGTA is coming, that you are creating a selection, and the selection by removing the non-euploid embryos, and then you don't waste time and money for those transfers. However, the question is what the data show. And I looked up the SART data from 2018, and that's maybe not the best approach because it's a little bit of mix of bags of everything, and not perfect. It's not a real study. However, it's also reflecting the daily reality, what we are living in. And those that are not living in the United States, SART is an organization that is collecting data from all or most IVF centers in the USA, that there are somewhat more than 400 IVF centers. And from 2018, what it shows that for patients who are younger than 35, the implantation rate was 46%. Those that had no PGTA, no biopsy. Now, in the same patient population, if they had a PGTA, then the implantation rate improved to 53%. For the patient age group 35 to 37, implantation rate was 41% without PGTA, and it increased to 55% after PGTA. Here at RBA, we looked at the same data, and we have seen that, in the youngest age group, less than 35, we had 53% implantation rate without embryo biopsy PGTA. That increased to 62% after biopsy and PGTA. Now, we can see that, yes, there is some improvement after biopsy and PGTA, but I think most of us would question, is this sufficient improvement? Is this what we expect? And I think most of us would think that no, if you have, let's say, 40% aneuploidy rate in the group of 35, and let's assume that you have 30, in the less than 35, you have 40% aneuploidy rate, and you remove that 40% of embryos from the group of embryos that you transfer, then I actually calculated what should be the expected implantation rate in this group, and that should be 76% and not 53%. And similarly, in the 35 to 37 group, it should be 82% the expected implantation rate and not 55%. So, and we got similar results also when we looked at our RBA data. So looking at, SART result is not the perfect way. Doing studies is a better way, to see what is going on really. And I just want to refer to one slide here from a study that was a multinational, multi-center randomized controlled trial, pretty well designed, not perfect, but pretty well designed, where they transferred just a single embryo either after PGTA or without PGTA, without biopsy. And that's a very recent study actually. And what you can see here in the patient age group of 25 to 34, the pregnancy rate, again, single embryo transfer, was 53%. And after PGTA, it was 49%. So actually, instead of improving the pregnancy rate, it seemed like a little bit of decrease. Of course, that statistically was not significant. In the patient age group of 35 to 40, they saw 37% pregnancy rate, ongoing pregnancy rate in the group of no PGTA, and 51% in the PGTA group. So here we can see a slight increase, although it didn't really reach significance that either. Now, this study data was used by another group who published their findings that they tried to establish that, what was the result of that study on the patients. And they have published this in Reproductive Biomedicine Online only just a couple of weeks ago, saying that if you did PGTA in this patient population in the STAR study, that led a reduction from 82 to 50% of the cumulative live birth rate for competent embryos. And, to go back to our example where the patient had 4 embryos and we got 2 babies out of it. Now, if you are deselecting the 2 aneuploid, then based on this study result as well as, the SART analysis, basically that means that instead of getting 2 babies after PGTA, you probably would get just 1 baby. So the question comes up, what is the cause of this discrepancy? Some would ask, okay, is it a technique-related NGS? And, I'm not expert. I'm embryologist, so I'm not expert on NGS. But if you talk with experts, Gary and others, they will say that this is a rock-solid technology, and I believe them. I think that's the great technology. So what else can it be? It can be, of course, embryo-related and also IVF lab related to see that, what we are doing as embryologists in the IVF lab. And, we know that culture conditions can determine not only how embryos are developing, but it seems that it also determines that, what is the proportion of embryos that have chromosomal alterations, depending on those culture conditions. And of course, also some of the procedures that we do in the lab, like biopsy or tubing, that can also determine. And actually, that was the main focus of this webinar. And those 2 points will be discussed in the next 2 presentations, which is, I think, extremely important that we try to have more understanding and try to do our best. I just wanted to refer here a little bit to remind us, what are the origins of chromosome abnormalities in the embryo. And there are 2 origins. One is that is what we call meiotic errors, which means that there is a chromosome abnormality either in the sperm or in the oocyte or in both. And when that happens, then the resulting embryo, in every cell of that embryo, you will find the exact same chromosome abnormality. So that's a uniform abnormality. During mitotic errors, and actually that happens quite frequent, which is a post-zygotic error, if there is a chromosomal abnormality arising, then typically that will be confined just for some of the cells, and it's not going to be present in all the cells. And here I just also wanted to remind everyone, and there is no time to discuss about it more in detail, about cell cycle and cell cycle checkpoints. Because what happens when the cell is dividing and goes through meiosis or goes through mitosis, there are cell cycle checkpoints. That means that when the DNA is duplicating and the chromosomes are matching up on each other, if there is any mistake, then those checkpoints, they say, hops, that's not good, and stops the cell cycle progression. So in theory, if you have a chromosome error in a cell, then it's not going to be able to divide. So unfortunately in human, that's not working very efficiently. And that's one of the reasons that a very high proportion of embryos have chromosomal abnormalities. However, sometimes it does work and stops the cell cycle and stops the cell dividing. And that's when you sometimes see, after collapsing a blastocyst, some larger cells in the periuterine space. So here I wanted to jump to the second topic that is related to, of course, to the topics that I have been discussing here, which is concordance after re-biopsy. And what we did here at RBA is that patients who had embryo biopsy PGTA testing and it came back as abnormal, 1 or 2 chromosomes abnormality, either full abnormality or mosaic and segmental, and the patients donated those embryos to research. Then we did total embryos, and depending how many cells the embryo still had, we took out some other pieces and then we analyzed those pieces. We tested a total of 79 embryos and a total of 213 samples. And basically what it showed, that 85%, 86% of the time, it showed a complete match of the re-biopsy sample to the original diagnosis, to the original testing. In about 13%, it showed a partial match, and in 2.6%, it showed a complete mismatch. Now, in this table, we did not include the mosaic or the segmental testing diagnosis, but when we included those also, the mosaics and segmental, then the complete match actually decreased to 65%. And there are a few publication studies about the same concordance question after re-biopsy. One is from Chang and Korix from Molecular Human Reproduction from 2018, where they looked at the 2 trophectoderm biopsy matching that was 90%, then the T1 versus the ICM that was 86% match, and the T2 with the ICM that was 90%. So their finding is somewhat similar to ours. Saktev and colleagues published in JARG this year, where they found that about on 32 embryos that 40% of the re-biopsy specimens were concordant for all chromosomes in the initial biopsies, and they found that it was higher, the concordance, when the embryo was euploid or aneuploid, but it was lower when it was mosaic. So in summary, we can say that PGTA may improve implantation rate per transfer, but it does appear to decrease cumulative pregnancy rates. Re-biopsy retesting of embryos show that there is not a full concordance with the original test results, especially in mosaic cases, and that the role of IVF laboratory to optimize outcomes after PGTA is really important. And the critical elements include embryo culture conditions, embryo biopsy and tubing techniques, and cryopreservation vitrification. That's a topic that we didn't really talk about. I didn't talk about it. There is no time on one hand, but it's extremely important because most of the time when we do biopsy and PGTA testing, that embryo is going to be vitrified and used in a frozen embryo transfer cycle. So how we perform, cryopreservation vitrification is actually very impactful on the outcomes and results. But thank you very much.
Peter, thank you so much for that that really good review of the data that you guys have. So we just have one question here so far that we have time to ask you. You mentioned cell cycle checkpoints and how they don't always work, certainly in early human embryos. So if those checkpoints are not working, mitotic errors would then be quite common in embryos, which I think was pretty clear, which would obviously lead to higher mosaicism, right? Because mitotic errors are where we get mosaic-looking embryos. Do you think mosaicism is a more common occurrence in embryos than we maybe previously envisioned? Is it maybe just, or do you think it's common in the trophectoderm and maybe not so much in the inner cell mass? And then lastly, what is your opinion on transferring mosaic embryos?
Wow, thank you so much. Okay, so trying just to be very brief, I think the the mitotic error in human embryos, I think it's higher than what we are probably expecting or what we are estimating. That's my own opinion. If you remember Yuri Verlinsky, who has very much advocated to do the gamete testing and not so much the embryo testing, because what he told basically, if you are able to test the gamete, and especially the oocyte, because most of the meiotic chromosome errors are coming from the oocyte and not from the sperm. So if you are able to test the oocyte and the oocyte is fine and does not carry chromosomal alterations, then you really don't have to test the embryo, because even if it has chromosomal alteration in the embryo, in absence of a gamete alteration, chromosomal alteration, then it's really just a mitotic error and not a meiotic error. So it's just a mosaic situation, and either the embryo is developing to a blastocyst stage and then you can transfer or not. So unfortunately, testing gametes is very challenging technically, so that's not really working. But altogether, I think my opinion is that mitotic errors are contributing a big part of the chromosomal abnormalities that we see and detect in embryos, in human embryos. Personally, regarding the transferring mosaic embryos, I am very open about it. I think that's if a patient feels comfortable with the idea and gets consultation with a genetic consultant, I think that's a good idea to do it.
Right. Thank you. That was a very concise answer to a very difficult question.
Thank you.
Okay, so we're gonna stop with we're gonna finish up with Dr. Nagy, and we're gonna move over to Dr. David McCulloh. David has the unique distinction of being a lot of different things in the embryology world. He's an embryologist. He's my personal statistician, which is a really good thing. He's the only person in the world that can say he's my personal statistician. David works at the New York University Langone Fertility Center, is the lab director at Biogenics Corporation and the Sperm and Embryo Bank of New York. He's directed art laboratories for nearly 3 decades and is the director of clinical science at ReproArt, Georgia American Center for Reproductive Medicine in Tbilisi, Georgia. Prior to his clinical endeavors, he investigated sperm-egg interactions using cellular electrophysiological methods in sea urchin eggs at the University of Miami School of Medicine and in rabbit oocytes in vitro at the University of Maryland and the USDA. David is a good friend, an amazing speaker, and I think you guys will really enjoy his talk today. So with that, David, to you.
There we go. Now I just unmuted myself. Hopefully that worked. You hear me now? Good. Now I'll share my screen. And let's see, share, and we will go to this. There we go. Okay, so I'm honored to have been selected to share in this presentation. So I thank Peter very much for the invitation, and it's been fun interacting with everyone on this. I want to talk a little bit about some places where ploidy errors may arise in embryos, and possibly places where we might be able to change the way we treat the embryos and the oocytes to avoid them. But first, where and when does aneuploidy occur? Peter went over this a little bit, that aneuploidy, true aneuploidy, occurs during meiosis. and it affects the egg, so all the daughter cells are affected. Whereas mosaicism, true mosaicism, if you can see my pointer, occurs during mitosis. It's post-zygotic. It affects daughter cells, and it may affect all or some of the cells in the embryo. And the point I want to make is that mosaicism, true mosaicism, occurs during mitosis while the embryo is in the lab, because that's when mitoses occur. Aneuploidy, we like to believe happens before we get the egg, but actually the second meiotic division can be a source for aneuploidy, and that oftentimes, well, that happens after the sperm enters, so that definitely happens in the lab. And occasionally the first meiotic division may occur in the lab as well. But I want to make one caveat before we go any further, and it's largely in response to what Peter presented concerning re-biopsy. There's some imprecision in the results of trophectoderm biopsies. Discordance upon re-biopsy suggests that there's mosaicism in the embryo. If the two biopsies don't agree, the embryo is mosaic, and yet the initial biopsy may have said it was aneuploid. So we have to realize that there are times when trophectoderm biopsy may result in an aneuploid result when the embryo is actually mosaic. So aneuploid results may not mean true aneuploidy. So I'm going to talk about the results that we've received from our testing and comparing aneuploid results and mosaic results in various ways. And I can't really discuss directly anything about true aneuploidy or true mosaicism. So the first thing I wanted to point out is that we can ask the question, is euploidy rate the same for all programs? And this was a paper in 2017 in Human Reproduction by Santi Munet and colleagues, including me. And we here have plotted 42 different programs that had their results run at Santi's labs in the United States. There were 1,600 or so donor cycles. There were over 13,000 blastocyst biopsies, and as I said, at 42 centers. It was looked at with array comparative genomic hybridization, ACGH, not NGS, but we don't need it at this point. And what we have here are these 42 centers. Each bar represents the euploidy rate for donors at that center, and they're in order from the lowest euploidy rate to the highest euploidy rate. The red bars are significantly lower than the average, which is around 69% euploidy rate. And the green bars are centers that had significantly higher euploidy rates than the average. So what we were surprised to see was that different centers had significantly different euploidy rates for donor oocyte cases, where we expected things would be quite similar. So if there are differences at different centers in a population where we would expect things to be quite similar, then where are they coming from? So the first question that comes to mind is, does clinical treatment contribute? And for a follow-up study in European Journal of Medical Genetics, we presented some data from one center that had 6 physicians. There were 423 donor cycles in one embryology lab using PGTA, it's ACGH, at one PGT laboratory. And what we found here at the bottom is that the euploidy rates across the bottom in this red box were significantly different when we compared the 6 different physicians. So they're all using the same lab, they're all using the same PGTA lab. What could be contributing to these differences? And we looked at a number of the cycle characteristics, and the cycle characteristics were all significantly different for the 6 different physicians as well. Then we decided, well, since we have these differences in euploidy rates and differences in these cycle characteristics, perhaps we can look at the cycle characteristics and the euploidy rates, not so much comparing between physicians, but just comparing between donor cycles. And when we did that, we found that there was one parameter that was significantly associated with the euploidy rate, and that was this fraction of the total gonadotropin that was contributed by HMG, human menopausal gonadotropin, the old style gonadotropin. What we found was that with higher amounts of HMG in the mix, we had higher euploidy rates. It seemed to reach a peak around 50% of the gonadotropin being contributed by HMG, and the total effect between 0% HMG and 50% HMG was about a 5% increase in the euploidy rate. So overall, our point was, there seems to be something that can be contributed by stimulation. We don't know all the things, it's not an exhaustive study of that, but we certainly were able to find something that suggested that the stimulation may contribute to the euploidy rate for the embryos. But the question that we really want to pursue today is, are there things that we do in the lab that might affect the ploidy? So that's what we're going to talk about a little bit here now. And what's probably the best place to look right now is a wonderful review written by Jason Swain. It came out recently, 2019, in RBM Online. And he looked at various different things and provided a really insightful and honest appraisal of where we stand with what we know about how things in the lab affect the embryo ploidy. We look at temperature. Well, you might expect the temperature would affect things. We know that temperature can affect spindles. And if it can affect spindles, you might expect that there would be an effect on segregation of chromosomes. Probably the best study was one that looked at static change of temperature, meaning that they cultured some embryos at 37 degrees, others at 36. And there wasn't really much effect on the euploidy rate. There was definitely an effect on the rate of development, but not on the euploidy rate. What wasn't really looked at was fluctuations of temperature and how they might affect things. And we all have kind of used fluctuations as a reason why we've made this transition over to benchtop incubators, which can recover much more rapidly from an opening. But this was not really considered in the review. Another thing to consider is oxygen tension. And there are some quite suggestive papers out there suggesting that oxygen tension may contribute to differences in euploidy rate, or actually mosaicism predominantly. But it's mainly suggestive. Again, there's also studies looking at culture media. This usually kind of falls out into things like comparing pH levels and osmolality. pH changes, of course, reflect or may reflect what the CO2 level is we're using for the gases above our medium. Osmolality may suggest the humidity in the incubator, or perhaps the kind of oil or thickness of the oil layer or heaviness of the oil that is overlaying our drops in culture. So these are all things that have been shown to have something to do with, or are in some way associated with ploidy in embryos, but it's predominantly suggestive. And he also discussed plastic wear. So there are definitely some things that are worth looking at, but it's not necessarily so clear exactly what is the cause of these differences in ploidy and mosaicism. Well, I'm going to try to illustrate a few of these examples from stuff that I had available to talk about. One thing that wasn't discussed in Jason's paper was whether follicle diameter is associated with euploidy. This study was performed in Georgia at the program I work with in Georgia. We looked at the diameter of each follicle measured at retrieval during retrievals from 22 different oocyte donors. The oocytes were retrieved from each follicle and cultured individually so we could track the follicle size along with the oocyte and maintain those things. They were all undergoing ICSI so that we could judge the maturity shortly after retrieval. The analysis of outcomes was done in relation to the diameter of the follicles from which the oocyte was obtained. And the observations were that follicle size is associated with oocyte maturity. That's not really a big surprise to any of us, I think, but I think it's a pretty clear illustration of the fact that the immature oocytes tend to come from small follicles. But what I was more interested in and what we focused on in the study was whether follicle size was predictive of whether an oocyte would become a euploid blastocyst or not. And what we found was that it didn't matter what size the follicle was, as long as the oocyte was an M2 oocyte, a mature oocyte. It didn't matter what size follicle it came from, it had an equal chance of being or becoming a euploid blastocyst. So what we found was that follicle size does not allow us to predict whether the oocyte will lead to a euploid embryo or not. Now, why am I discussing this as a lab function? Well, the reason is sometimes we flush a follicle and we don't get an egg. Well, I think it's important to realize that it could be any size follicle that we're flushing and we don't get the egg from. If we don't get the egg and it happened to have been a mature oocyte, it had an equal chance of being a mature oocyte that would lead to a euploid embryo. So we can't just say, oh, don't worry about those small follicles, or don't worry about those big follicles. Doesn't matter what size the follicle is, the oocyte, if it's mature, has an equal chance of becoming a euploid blastocyst. Next question, does oxygen tension affect ploidy, or is it associated with ploidy? And this was a study, a brief study, looking back retrospectively at data here at NYU. Between the years of 2015 and 2020, we looked at about 4,400 cycles. We were comparing between incubators, those with ambient O2, these were all big box incubators, and those with low O2, some small box incubators, benchtop incubators, and one that had a video camera in it. And we can compare the euploidy rates here between embryos that were incubated at ambient O2 levels, around 20%, and those incubated with low oxygen levels, 5%. We find that the euploidy rate was significantly higher in those embryos incubated in low oxygen. Likewise, mosaicism led to higher numbers of embryos being mosaic in the high ambient oxygen levels and lower mosaicism levels in the low oxygen. So it suggests that there's something going on here. It's not clear that this is the only factor going on, but it certainly suggests that it's preferable to be incubating in the low oxygen level, preferable over the ambient oxygen levels. But another thing that we wanted to look at was an examination of ploidy as an outcome when we compare the personnel that are handling the oocytes and embryos. Now, this is kind of a long shot. You have to bear with me, and hopefully I can make it clear. We're looking at PGTA results, euploidy and mosaicism, and we're comparing different technicians who've handled that oocyte or that embryo at different times during the treatment. We're looking at the inter-technician variability in this evaluation. And we look at the embryologists that perform different embryology procedures. We look at the euploidy rates and the variability of euploidy when comparing different embryologists that performed retrievals. Or we looked at the euploidy rate when different embryologists performed insemination, or when different embryologists performed the biopsy. And we're looking predominantly at those situations where the variability between technicians was large or larger, and those stages where the variability between or among embryologists was small. So we're comparing stages, the stages when we do these procedures, and trying to find those stages where the embryos are more susceptible to the effects of handling. So if there's more variability from embryologist to embryologist, we suggest that that particular stage is more susceptible to handling than other stages where the variability is not as great. The general information, there are over 4,600 cycles with PGTA. In this case, we're using NGS, and all of it was done with trophectoderm biopsy. We compare some things having to do with sperm exposure. There were 2,700 cycles with insemination, 1,200 with ICSI. Cycles with both were excluded because it was a little harder to figure out. And there are roughly 22 technicians that were being compared over these several years of data. Here what we have plotted is this standard deviation among technicians. Now this is the inter-technician variability on this axis. And here we have different stages when handling occurred. Retrieval, sperm prep, insemination, ICSI, FERT check following insemination, FERT check following ICSI, zone of perforation, and biopsy. We do our perforations predominantly on day 4 now. And then the biopsy. And what we see is that there's quite a bit of variability in this inter-technician variability. Insemination, standard insemination, seems to be the time when there's the greatest variability between technicians. So I would infer from this that there's a likelihood that this stage is particularly susceptible to handling, and we may want to start focusing on that. The fertilization check is the time when things are the least variable, and these are all in comparison to this line, which is the within technician variability. So there's something about this time which suggests that the embryos are much less susceptible during this period of time than they are before, which these are all before meiotic divisions are completed, and these are all after meiotic divisions are completed. And yet we're still seeing what appear to be effects on the euploidy rate, but as I mentioned earlier, euploidy may be detected with the result, but it may actually be a mosaicism that we're just failing to detect the mosaicism. Then the final thing I want to look at is, again, a similar comparison, but here instead of looking at the euploidy rate, we're looking at mosaicism. And again, the early stages prior to meiosis completion seem to be more uniformly susceptible to effects of handling. Again, fertilization check is kind of low. Zone of perforation is a little bit high, and biopsy is not particularly high. So we have differences. I'm wondering if, I don't really know quite how to interpret this, and I'm actually presenting it because I'd like to have people provide what their feelings are about this. But it does appear that there are differences at different times. In summary, we know that age affects euploidy. We've seen that in a myriad of papers. What hasn't been so clearly seen is that actually mosaicism is not so dependent on age, although it seems to be thrown out there quite often, that it isn't particularly dependent on age. Controlled ovarian hyperstimulation, at least the use of HMG seems to be associated with a small but significant effect on the euploidy rate. And in the laboratory, there are many features that are associated with rate of development. That's pretty well reviewed in the J. There are many features associated with ploidy, among them oxygen tension, which I showed you. Handling of the oocytes by technicians appears, at least in my eyes, to be something where we might be looking at a way where we can start focusing on how to improve. But I think I want to sort of throw in another caveat, and that is that lots of these things that we've seen could be affected by many factors that we're not really looking at. We focus on one particular feature and say, well, that's causing this difference in euploidy rates or mosaicism rates. But in fact, there's so many factors going on that it makes it very difficult to identify specific features. And so with that, I'll stop and say thank you for the opportunity, and I hope I may have stimulated some questions.
Dave, thank you so much. I really appreciate it. Really, really detailed, as always. You can always count on Dr. McCulloh for a really, really detailed question with lots of information and data. Okay, so I have just time for one question for you. It's a really easy question, Dave. It should take you no more than 3 seconds to think about it. how do we as a community of embryologists tackle quality control amongst embryologists handling techniques between the labs? It's a pretty simple question.
Yeah.
Not
Well,
at all.
Well, I think it is actually relatively simple. And a few sessions ago, we saw Joe Conahan present information about how he was comparing the blast formation rates between cases that were performed by his technicians. And he found that some technicians performed better and some technicians performed less well. He then showed that he was able to adjust and improve his overall outcome by having the technicians who weren't doing it the way the best technicians were doing it, do it the way the best technicians did it. Right. I think we can do the same sort of thing if we look at euploidy rates and mosaicism rates, comparing between technologists that are performing different particular steps in the whole procedure. But what makes it a more huge task, I think, is that we seem to be seeing effects at very different stages. It isn't just at time of ICSI. Even sperm preparation was looking as though it was one of the larger contributors to differences in euploidy. So it raises some interesting questions, doesn't answer many, but I think it raises questions, and I think the question that was asked is right on target. And I think if everyone starts thinking about the question, they can come up with ways to try to address how to approach this in their own labs.
Yeah, I think it's incumbent upon anybody that works in a lab or runs a laboratory to do proficiency testing to make sure that there's no drift. I know I've spent a lot of time in my labs doing proficiency testing, external, internal quality assessment, those kinds of things. And I think those things fit in really nicely here as well. Thank you very much, Dave. We appreciate it.
Thank you.
So with that, we're going to switch over, and I'm going to introduce my friend and colleague, Dr. Dawn Kelk, who received her PhD in 1997 and her HCLD in 2000. You will hear a little bit of that Canadian accent when she speaks. She has directed labs both in Canada and the US. She's currently the director of the laboratory at Yale Fertility Center and is an associate research scientist at the Yale University School of Medicine. Prior to that, she was the lab director at RMA of Connecticut and the University of Colorado. And if you've never heard or seen Dawn talk, you're in for a real treat because Dawn really, really knows her stuff. I'm really looking forward to this talk. And with that, Dawn, I'm going to hand it over to you.
I just forgot to unmute. Can you hear me now, Gary? Okay. Well, I have to say that that's the first time I've been introduced by somebody with a mohawk. So I appreciate that, Gary. And this has been a lot of fun getting involved with this group. All right. So I'm going to do just a little bit of rehash over what Dave McCulloh had mentioned, and maybe just illustrate it in a slightly more graphical way. I think Dave did a great job of sort of explaining that sort of what we know and we don't know. We know that meiotic errors lead to aneuploidy, and those are at the oocyte level. So this is, oh, sorry, jumping around a little bit. My mouse is being a little funny. All right. So we know that aneuploidy is maternal age related, both in vivo and in vitro. We know that miscarriage rates are higher for in vivo pregnancies with older women, and we know the same is true with in vitro. So we do know that aneuploidy is an egg phenomenon. We also know that mitotic errors lead to mosaicism. So this is just a little illustration. As we have cell division happening, if we are mishandling an embryo somewhere along the way and with maybe perhaps a temperature change, we can induce potentially a mitotic error, which would then be passed on to subsequent generations of cells. And we spoke a lot about this paper, and I think this is an important one to sort of touch base, that we know that euploidy rates in donor egg cycles do significantly differ between fertility centers. And this was an incredibly sobering paper for me, because that's an incredibly broad euploidy range between centers, from 39.5 up to 82.5%. So what's causing these differences in euploidy between clinics? Is it an egg issue? And if it's truly euploidy, which is touching again on what Dave McCulloh was saying, then it should be something to do with the egg, more in the range of superovulation, stimulation protocols, recruiting eggs that would not normally be ovulated. You know, we could be having some post-mature eggs. I'm really intrigued by the FSH to HMG ratio. That also impacts progesterone elevation at the end of the cycle, so it might be intriguing to tie all of this together as we go further. Certainly egg and embryo handling, with the spindle being incredibly temperature sensitive, and we know that there can be rapid changes with minor temperature fluctuations and pH changes. Or do we have an artifact of the biopsy itself and the testing? So to delve into that a little bit more, and I think this sort of illustrates what Dave was discussing, that when we have mosaicism, there's a broad spectrum of mosaic embryos that could have any range of diagnosis. So this first embryo, if we pulled all normal cells, the green cells being normal, we would diagnose this particular mosaic embryo as euploid. Conversely, the embryo at the bottom here would be diagnosed aneuploid, but would truly be mosaic. And I think that's what's getting into some of what Dave's trying to discuss, that the results that he's seeing are the results from the biopsy, not necessarily the embryo itself. So, so just a little bit of a graphic representation. So is mosaicism normally occurring, or is it a lab-induced, or is it an artifact with testing? And so I think this starts to go back to Jason Swain's paper, which David introduced as well. There's so much more to culture medium, and in the early days, you know, when you would go to ASRM, everyone would ask, what culture media are you using? What culture media are you using? As if that was the, you know, the special sauce that everything revolved around. And it was all about the culture media, and the media companies were the first ones to be thrown under the bus if somebody's pregnancy rates tanked. So now we know that there's a lot more than just culture media. It's a whole culture system. And within that culture system, we have to consider things like plasticware, the size of the drops that we're culturing in, the number of embryos that we're putting in those drops. Are we doing single embryo culture? What's the relative size of the embryo to the drop? Whether we're doing an oil overlay and how protective that is for preserving pH and preserving temperature as well. Certainly, we know that CO2 impacts the pH, and that's obviously a big one. And finally, the oxygen, and certainly low oxygen. Everybody's pretty comfortable with saying that low oxygen is definitely the way to go. Now, within that culture system, though, that culture system exists. If we sort of shrink that culture system down, well, first of all, actually, before I say that, all of these factors can interact together, not just necessarily independently, but there can be cumulative effects of all of these different items. Now, when we take that culture system lives within a laboratory. So here we have our culture system with all the different factors, and again, by no means comprehensive, because I think it was Rusty Poole came up with over 200 different variables that could go into producing an embryo through in vitro fertilization. So within the laboratory that culture system exists in, that culture system is also subjected to the air quality from that lab, from the various incubators that we could be using, whether they're benchtops or big box, the various equipment, the micromanipulation equipment, the refrigerators, and the benches and the isolates that we might be using, and of course the embryologists. Now that laboratory, again, can have all of those factors interacting together along with the culture system to produce impacts. Now that laboratory also has external factors coming in, the patient, the stimulation, the retrieval, the gamete quality, how the embryo transfer is performed, and the luteal support. And again, just scratching the surface on the really most obvious ones. So maybe one of the more important questions should be, how do you use your culture media, not what culture media do you use? And I wanted to just mention this quick example because within the Life Global media series alone, there are two different types of media. There's global and global total. And if you want to get the same pH range with global media, you need to use a CO2 of approximately 6.5% to get the same pH around the 7.222, 7.2345 in there. You need to be at 7.5% with global total. So again, how we use those media is really important in that culture system. So Dave already mentioned this great paper, which is a wonderful review. So I'll push on from that. Within this review, Jason Swain mentions the various stresses and susceptibilities that embryos have to stress. And all of these various stresses impact the embryo. We know that eggs are incredibly susceptible. The one caveat that I would say, and I think this reinforces nicely from Dave's, is that we also know historically that 2PNs were actually quite stable. So 2PNs were the stage that we would freeze at when we were slow freezing. They survived very well, so they were relatively stable, as opposed to the early cleavage stages where they're a little more susceptible. We've got more cells that can impact. And those cells we saw when we slow froze, if we slow froze an 8-cell embryo, frequently we would have 1 or 2 cells not survive. So mosaicism by biopsy embryologist varies a lot. So when we start to tease out some of those different factors, one of the challenges that we had, if we wanted to look at the laser situation, how do you design a controlled laser experiment with material that's highly variable from a limited number of embryos for research? And what we did in our group was we looked at some human embryonic stem cells, and we targeted those with a laser. We had control cells, cells that we would subject to a normal sort of moderate pulse in our lab use, and then we cranked the laser all the way up to maximum power. And what we found was that there was no change in the euploidy or mosaic rate in the biopsy samples. But I want to caution, because I don't think that means that you should go out and just gregariously go crazy with your laser use. Because when we were hitting those cells, we were definitely destroying them. The cells literally would go brown and atretic looking with the maximum pulse hits. So what you're doing to the remainder of the embryo, needs to be thought of. But the important thing was, from the biopsy perspective, the laser use was not inducing artifactual changes. And Frank Barnes has done some of the same work, and he didn't find anything when they were targeting with the laser on their actually embryo material. So the question is, when to biopsy and when to cryo? Do you wait for the embryo to prove itself? Do you wait for more cellularity? Do you consider the cryodynamics of an early versus an expanded or a hatched blastocyst? Certainly the fluid content is a consideration if you're freezing a fully expanded hatched blastocyst. Maybe not so much if you're collapsing those blastocysts before you freeze them, before you bit them. And certainly you're balancing handling concerns if you're dealing with a completely hatched blastocyst. I just wanted to mention quickly, particularly in the COVID days, that micromanipulation setups, I'm still amazed that there's still a fair amount of mouth pipetting going on in the world when I go out to different labs. And I really think there's not a lot of need to mouth pipette anymore. We've got some great various tools, and I think it's worth, I just put that plug in there. There's a lot of variability in the size of the biopsy pipettes that different labs are using. A lot of labs are still using 30 to 40 micron biopsy pipettes that were really designed for blastomere biopsy. And depending on the technique that you're using, that might or might not suit you well. In our hands, a smaller biopsy pipette in the 20 to 25 micron range facilitates easier trophectoderm biopsy. So one of the things we wanted to mention as well was that laser settings are important, and there's different kinds of lasers, fixed and static, all the various brands of lasers, and that it's important to target appropriately. And there's a lot of variability between labs with the laser pulse. So this is just a summary of what you might want to see in a lab. You would want to see this dish. Hopefully most people are familiar with what that dish would be sitting there. It's basically just a Petri dish with some dry erase marker on it. And when you hit with the laser on that dry erase marker, you can see the increasing pulses from 1 to 2 to 3 to 4 to 5, all the way up to 10 millisecond pulses. If you go down to this one, and I've certainly been to labs where the laser is just not functioning properly. And if your laser's functioning like this one, obviously there's an issue because you're not targeting properly. Hopefully your laser is going to line up so that you're appropriately hitting your target. If it doesn't, then you need to get into your settings and explore around a little bit. So I thought we'd have a quick look at some, I'm seeing a different screen than you. Oh, there it is. Okay, it was a little slow kicking over to the trophectoderm. So this is a typical trophectoderm biopsy pull and cut. So again, just pulling and stretching, typical standard biopsy that most people would do. The reason we like those smaller biopsy pipettes is because it plugs the hole a little bit better and the biopsy doesn't shoot up the pipette. This particular biopsy is just a different way of looking at it. These embryos obviously have been hatched ahead of time. These ones were hatched on day 2. What we did with this particular embryo was sort of turned it on its side to do the biopsy from the side, which you'll see. And it's still the same pull and stretch and cut with laser to pop that biopsy. But rather than pulling straight out, it gave us a little better purchase on the embryo to be away from the inner cell mass. So trophectoderm biopsy protocols we know can affect clinical outcomes, and this is a paper that came out fairly recently looking at whether you should pre-hatch embryos on day 3 or hatch at the time of biopsy. And this group was getting better results from not pre-hatching and giving the embryos that extra exposure. And the one thing they did do was they removed about 1/4 of the zone in the breech at the time of biopsy. So here's a quick look. And this particular video was shared by Peter Nagy. One of the nice things you can see is when he takes the hit here, you can see he's getting a nice clean cut on the zona, so his laser's nicely focused. It's going to pop in, grab some cells for the biopsy. And so that one was done without any pre-hatch. It was just collapsed and then breached at the time. So it was pre-collapsed, but not pre-breached. All right. So what do we know about different biopsy techniques? In this particular study, they looked at the pulling and pulling technique versus flicking. And this is just a small study from ASRM last year showing that the pulling and flicking methods did not differ in terms of rates of chromosome abnormalities. So I wanted to have a quick look at the flick technique. So The pressure is released on the blastocyst to allow grabbing a hold of the cells. And aspirating some of the cells. Lining up on the holding pipette. And I know there were a few people in the audience doing this technique. It looks incredibly traumatic and scary, but it does seem to be very effectual in the right hands. Okay, quick video now on what we would do at Yale for loading buffer into the PCR tube. We like to drag it up the tube just a little bit if possible. It allows a little better visualization. We don't want it too far up the tube because obviously the cell needs to be in the bottom of the tube. And then to look at biopsy washing, what we found is there's sort of what the testing companies recommend, which is passing the biopsies through drop after drop after drop. And then there's the reality of what actually happens in all the various labs. So one lab I was in actually spotted down 14 different 8 microliter drops to rinse through. So this was for one biopsy, second biopsy, third biopsy. So 3 biopsies were washed through the same dish with a couple of like drops to rinse your pipette. That particular dish, by the time it was spotted out and those 3 biopsies were rinsed through, was out for over 10 minutes. And you can imagine the osmolarity of those drops that are relatively small is probably evaporating. So again, a lot of variability between labs. This is my true confessions. This is how we do biopsy washing at Yale. We don't pass through all those different drops. We use a much larger drop, 50 to 100 microliters. And we'll pull the biopsy. This is just cleaning up the drop a little bit. But you'll see, this is Jonathan actually, going in. Jonathan's gonna pick up the biopsy and do some pipetting up and down to rinse it from the top of the drop to the bottom of the drop. So it gets a fairly thorough rinse, but it's not rinsed from drop to drop, where you're a little more likely to lose the biopsy because the biopsy is so small going from drop to drop. Okay, this is a visualization of the biopsy loading into the tube. So what you'll see is the pipette coming in here. And it'll come back on the screen just a little bit better. But if you watch closely on the tip, you'll see the biopsy exit the pipette right there. So we like to visualize that as we're loading the cells. It just gives us the confidence to actually see the biopsy going into the tube. And I know there are labs that don't do that. They just shoot the biopsy in there and don't visualize it, but we like to see it. So here's a paper from Foreman et al. Looking at fertilization method with IVF versus ICSI. I wanted to mention this quickly just because it's becoming a lot more popular, a lot more acceptable, certainly in the US, to be using IVF for PGTA. So this is reassuring that the findings on this paper are that IVF and ICSI, NGS PGTA had similar rates of euploidy and aneuploidy and no result embryos. So chance that they might have a slightly higher rate of mosaicism, but we'll have to get some more data on that. So in summary, I think it's a good opportunity for us to reflect on whether we're causing harm with PGT. And maybe perhaps PGT is our opportunity for introspection to be better clinicians and embryologists. Maybe it's actually showing us where we're making our mistakes in the lab. And so I'm really excited about Dave McCullough's data as we start to ferret out a little bit more what we're doing as embryologists and where we can be better. So I think we're really opening a window for insight into our culture systems, which I think is just going to benefit us. So I would hate to see us not continue to pursue PGT. So I also think that it's important for us all to remember, even though we're embryologists, to not lose sight of all of the patients that are having miscarriage, the miscarriage rates. For those older patients that are not doing testing, the time lost for a miscarriage is considerable, and there's a considerable emotional burden. So I think we need to continue to move forward with this technology, and the technology is rapidly changing, so it's exciting times. And I think that the future of non-invasive PGT is exciting as well. So, and with that, I'll turn it back over to you, Gary and Shaista.
Thank you so much. That was a fantastic presentation. And I think seeing these different biopsy techniques in action are an invaluable tool for many of us. So I'm just going to ask you one question from the audience. This question was asked by you, Sun. How does the invasiveness and the integrity of the nucleus of the retrieved tetraspectoderm cells impact biopsy results?
I've always wondered, and I don't think there are a few studies out there looking at whether they think they classify it as a good biopsy or a messier, more difficult biopsy. Those things are really difficult to quantify. So I think we just don't know yet. I think there's a lot more that we need to continue looking at and try and quantify this stuff.
Don, just for my side, I'm not going to ask you a question. I'm just going to say I was really happy to see you highlight the tube loading. I think that's an often overlooked part of what goes on the embryology side. You know, I always think of this as a collaboration between the genetics lab and the IVF lab, right? And if we can't do our job unless you do your job really well, right? And it's a really tough job. And so I think it's, for me, in doing biopsy courses, it's something that we don't often think of. Loading the tube is just as important as doing a good biopsy, right? You can ruin everything you did with a good biopsy by not loading the tube really well. So thank you for showing that.
I do feel like we don't always talk enough back and forth between the embryologist and the geneticist.
100% agree.
Because, you know, it may be that the genetic testing labs are recommending things that are a little more impractical or perhaps unnecessary. The one thing that I would like to ask you as a geneticist or anybody else on the panel is what you think about shipping biopsies with dry ice or with, you know, obviously we're shipping with freezer packs. In my lab, we started, because we've got a -80 freezer available, we use freezer packs that have been in the -80 freezer for what it's worth. Do you think that's helpful at all?
Yeah, I mean, I think, you know, every lab has their own thing that they like to do. Obviously, you know, you don't worry about anything melting or thawing when it's on dry ice, right? So that's the best. But dry ice, let's be realistic, it's a pain, it's expensive, it doesn't need to be done. I think, you know, for routine work, depending on where you are, like for you, if you're sending your stuff to California, it's different than if you're sending your stuff to New Jersey as well, right? And always, obviously, it's not the 80% you can control, it's the 20% you can't. That when things go wrong, you want that sample to be as cold as it possibly can, right? When it gets lost by FedEx or whoever. So I think for routine work, honestly, you know, when I was at iGenomics, we designed a special packaging box, and we used just frozen ice packs, and 99% that worked perfectly fine, and it's much easier on your end than it is to try to pack dry ice. I've had conversations as well about, you know, the integrity of the cells. They're getting ripped out of an embryo. You know, I always feel like you should keep it as cold and or at least as cool as you can through the whole process. I've had embryologists disagree, said, listen, we do it at 37 degrees and our results are great. So, you know, clearly I don't know everything there is to know about this. I have my opinions. Certainly these cells have just been ripped out of a growing embryo. They can't be very happy. And so we know from molecular biology, the cooler something is, the less cellular processes that are going on. And when things are unhappy, the cellular process that's going on is apoptosis and shearing of DNA, which is never a good thing for us. So
right.
You know, that's not an answer to your question, I don't think, but I think that's something to think about when you're thinking about tubing and shipping is, you know, controlling the things you can't control or where the rubber is going to meet the road.
Well, and that just reminded me that I didn't actually mention it in my talk, but one of the things that we do that Nathan Treff put me on in the early days when I was with RMA was to use a cold block. So we have a small, basically aluminum PCR block that we put those little PCR tubes. And if you noticed in my video, when I first focused on the PCR tube, when we were loading it, there was a little bit of condensation on the back of the tube that quickly evaporated so that you could visualize. But we do, when we load the buffer in, we put those tubes in that cold block.
Yeah, you could also use, Kelly Ketterson turned me onto this one, which is a frozen ice pack with a chuck or a Kim wipe over top of it, and then set your PCR rack on top of that. It's not perfect, but it will work. It will keep things cold. And also, again, not that this is a commercial for iGenomics, but that's the last time I worked in a lab. We use those frozen PCR racks that change color as they warm, right? So you can tell where that rack is warm and where it's still frozen, which is, again, I think it's easier for the embryologist to understand how long things actually take and how things are changing through time. Okay, so we have a final poll, and we're going to see if anything that we said today has changed the audience's opinions on the matters of PGTA. So while you guys are doing that, I'm going to thank myself and Dave and Dawn for the presentations, and Peter. And we're going to open the floor now to general questions. Please, again, use the Q&A function. We've got some good questions in there. And so we're going to go through. We don't have a ton of time. We're going to go through, and each person's going to get one more question asked while you guys are doing the end of the session poll. So, Shaista, you want to kick off the first one?
Yeah, I can ask the first question asked by Estrella Rosenberg. Is there something you do in your biopsy that increases the noise of the sample, and then the result is that the embryo is classified as abnormal? And I think this question is a general question for all our panelists, so feel free to chime in.
Can I start?
Absolutely.
I'm not an embryologist, Peter, so you can go ahead all day.
Okay. So first of all, I don't know what is the right answer, but I do believe that, for instance, if you are selecting an embryo that has more cells, it's better than if it has less cells. Also trying to avoid to take with your biopsy pipette cells that are larger sized, because that cell might be a abnormal cell just because it's larger. Obviously you could try to avoid going through in places if it was conventional insemination that you could get a sperm in it, although most people, they say that sperm is not going to get amplified, not going to influence the result. The same way, if there is still some attaching corona cells around the zone, also try to avoid it. So I think altogether, if you follow those precautions, I think you are at the safer place. I remember when I started, doing embryo biopsies 35 years ago, we changed biopsy pipette after each embryo. So I don't know if, that makes a difference, but, we were extremely precautious back at that time.
Thank you, Peter. I think that's an excellent answer. Gary, do you want to ask our last question?
Sure. So this is from Mike Large at Cooper. So do any of the speakers have any hunches about the likeliness or likelihood that the many variables associated with growing blastocysts may have an impact on non-invasive PGTA? We didn't really talk much about non-invasive. Today was all about, well, I guess, mostly about invasive PGTA. Certainly my talk goes, it's the same concept, NGS for non-invasive or invasive. Mike, I think I'll start. I think non-invasive is really, really interesting. If you haven't seen it, the iGenomics group, Carmen Rubio has a preprint of their trial out recently, which is quite interesting. For us, from my company, certainly when we developed our non-invasive kit, there's a great many things that go into embryo media that are not necessarily good for molecular biology, right? Salt, EDTA, protein, there's potential DNA sources in there. So there's a great many things that go on in the embryology lab that will definitely impact your concordance rate with non-invasive PGTA. I think non-invasive is really cool, but I think we have a lot of work to develop some data to truly understand what non-invasive PGTA results mean for the end user patient.
Since we are very short on time, thank you all for sticking with us. I know we are over an hour and a half into the session.
Yeah. All right.
Any chance to see some of the polls answers?
Yeah, that'd be good, Thomas, if we could do that.
Okay, so what is the effect of PGTA on the per transfer pregnancy rates? 45% said it improved significantly. What is the effect of PGTA on the cumulative pregnancy rate? 36% said it improved significantly. To which patient would you recommend doing PGTA? And 74% of you said more than 35 years old.
Yeah, it's
Those
really
are
interesting.
Yeah, it's very interesting.
I wish we had more time. Well, so first off, I want to just thank everyone for having me. It was really fun. It was really good talks. I really enjoyed it. Thank everyone that joined us today for the great questions. Again, it's like Shaista said, for sticking with us for so long. And to the panelists for the informative discussion. I also need to take a quick minute to give a shout out to the founders of this initiative, particularly Dr. Jacques Cohen and Dr. Peter Nagy, Thomas Elliott, Shaista Sadruddin, Marianne, and I can never say your surname, Marianne, so I'm going to just leave it as Marianne. Thank you for the vision and ambition to start this invaluable platform. Audience, please join me to give a gigantic virtual applause to our founders. So thank them very much. I'd also like to encourage you again to contact us if you'd like to help create or submit some website content through our website, or email us at contact@ivfmeeting.com. If you represent a professional organization, please help spread the word. Let us know if you'd like to host a session on your own topic, even in your own language. We aim to have this operation active continuously to maximize the value to our community, not just during COVID, but I think after COVID. I think, you know, anytime you can educate the field and get education for yourself, it's a fantastic thing. Okay, we have one more poll question that I'll go over really quick. Has your opinion on the use of PGTA changed after seeing these presentations? 55% of you have said that your opinion has not changed, and 37% now think that it is more beneficial. Meanwhile, like 10% think that it is less beneficial. I think there's still a lot to unravel in the whole realm of PGTA. So I think having these conversations and these engaging discussions is very crucial to that. So thank you once again to our 4 speakers. I would also like to give a big shout out to our audience for showing up at all odd hours of the day and for asking fantastic questions to keep the conversation so stimulating. We appreciate all of your support. Just a reminder that the recording will be available on our website shortly. And if there is more interest in this topic, we can revisit it in a few weeks. The 22nd I3 session will be held at the same time and same place next week, Tuesday, June 23rd at 8 p.m. GMT, 9 p.m. CMT, and 3 p.m. EST. The session is entitled, Why Does Osmolarity Matter? Unwanted Changes, Explanations, Observations, and Countermeasures. Our moderators will be Kim Pomeroy and Brian Lamonto. We will have 2 fantastic presentations by speakers, Dr. Stephen Mullen with Cook Medical, who will be sponsoring our session, and by Dr. Jason Swain, the laboratory director over at CCRM. We are excited to have such a highly esteemed array of scientists presenting on such an important and sometimes overlooked aspect of the IVF lab. We hope to see you all there. So with that, I'll say thank you to Shaista, because you were a great co-host. You did a great job. I wish everyone a wonderful rest of your week. And in this odd time of COVID, be safe, stay healthy, cut your hair funny. It does make you smile. It makes everyone else happy around you. And stay safe. We'll see you next Tuesday. Thanks, guys. Everybody. Bye.